Okay, hello, hello. Um, the I don't know if people see me. My name is Mathias de I3h, and I'm very, very happy to introduce our speaker of the day, Ilaria. Also very happy to see Hilde, full of energy and about to become more and more mobile after her intervention. So it's great. We are with you. So extremely happy to welcome Ilaria in particular because Ilaria is I3h's first PhD. I3h and okay, also ECARES, of course. So and she went through the ECARES doctoral school. But clearly, I think she embodies a number of things that are very good in this center I3h. In particular, the interaction between life scientists and economists, and in fact, the we had started working some years ago on the opioid crisis. I had suggested to Michel to look at this because I had heard it was so bad in the US. I made a presentation here of not of an original paper, but of some reports and so on this. And thanks to Michel Goldman, we managed to get access to a French database on opioids. And with Ilaria, with her supervisor, Patrick Legros, with Michel, with myself, with Victor Ginsberg, we started working on this. And in fact, it became a successful paper on the opioid and so-called death of despair. The title of the book, a famous book by Anne Case and Angus Deaton, who had just received the Nobel Prize for some other work. We had a project on that, which turned out to be a paper in the European Journal of Health Economics with Ilaria as first author, which in economics is not typical. We go alphabetical, except when indeed one person does so much, which was this case. You've gone on to do other things. Also, very interesting, that landed you a job as postdoc in the famous Toulouse School of Economics, where Pierre Dubois is helping to coach you, head of the Toulouse School of Economics Health Center, also with Jean Tirole being around and so on, which never hurts. Anyway, in the meantime, you have produced a very nice paper on the impact of pollution and antimicrobial resistance. And we've heard it already once here at the ULB, but that was an ECARES conference in honor of the retirement of Patrick Legros. But anyway, I'm sure the paper, which was already very good, has even further improved, and we are very happy to listen to you.
Amazing. Thank you very much for a great presentation. I'm indeed very happy to be here today and to present my latest piece of research that indeed studies how air pollution favors the spread of antimicrobial resistance in EU countries. So let me start by asking, why should we care about antimicrobial resistance? Well, because on average, every 7 seconds, someone gets infected by a resistant bacterium. And this can lead to reduced productivity at work, decline in workforce participation, hospitalization, and even to death in the most severe cases. Indeed, antimicrobial resistance, or AMR, is already causing about 35,000 deaths every year in the EU alone, imposing an economic burden of about 1.5 billion euros, again per year, and again in the EU alone. The World Bank considers AMR as a threat to the global economy that could cause the global annual GDP to fall by up to 3.8% if no action is taken, so in the absence of effective policies. Now, traditionally, we have been blaming the excessive use of antibiotics for this problem. So we think of antibiotic overconsumption as being the main responsible for rising AMR rates. And perhaps surprisingly, little is known about other potential risk factors. However, recent scientific research suggests that antibiotic stewardship programs, so reducing antibiotic use alone, will not be enough in the future to curb AMR. And this is because the transmission of antibiotic-resistant bacteria and genes through the environment represent, in fact, a primary driver of AMR. One potential environmental pathway is given by the air. So this recent scientific research has found that air pollution carries important quantities of antibiotic-resistant bacteria and genes. This can be the result of processes such as evaporization or aerosolization. One hypothesis is also that air pollution favors horizontal gene transfer. So we know that bacteria have the ability to transmit genetic material to one another, including resistant genes. And this process is indeed known as horizontal gene transfer. So based on this recent scientific literature, this paper is the first to provide causal estimates on the impact of air pollution on the diffusion of AMR. I do this by focusing on EU countries and the period 2002 to 2019. I also specifically focus on fine particulate matter or PM2.5 concentration as a proxy for air quality. This is because this specific pollutant is found to be the most harmful for human health. So is predominantly responsible actually for the adverse effects associated to air pollution. And this is because this pollutant is especially thin. It's about 1/4 the diameter of a human hair, and therefore it has the ability to penetrate deep into the lung and so to transport, to potentially transport antibiotic-resistant bacteria and genes. So, of course, air pollution is not randomly assigned across geographical areas. Two potential sources of endogeneity for air pollution that are frequently mentioned in the health economics literature are residential sorting and avoidance behavior. So let me give you the example for residential sorting. Suppose that all individuals in lower socioeconomic conditions tend to reside, to live in the more polluted areas. Now, we know that these individuals typically are more vulnerable, they have worse baseline health, and therefore they are more likely, they are more prone to get sick. So when studying the relationship between air pollution and AMR, we may be observing a positive correlation between the two just because of these underlying mechanisms. Okay, so what we want to do in these cases is trying to isolate the causal effect of air pollution on AMR. And what we frequently do in economics is we try to find another variable that is exogenous and affects air pollution, so that causes exogenous variation in the quality of the air. This is called an instrumental variable. So in this paper, I established causality indeed by exploiting an instrumental variable that uses the occurrence of temperature inversions as excluded instruments. So temperature inversions are atmospheric phenomena that occur when a mass of warmer air keeps pollution trapped near the surface. So let me explain you this better with the help of some graphs. Yeah, so under normal atmospheric conditions, which would be the graph here on the left, we know that there exists an inverse relationship between altitude and temperatures. So the more we move up into the atmosphere towards higher altitudes at lower pressure levels, the more temperatures decrease. When a temperature inversion occurs, this relationship is reversed, so that the mass of warmer air literally keep the smog trapped near the ground. Now, to give you a real-life example of this, here you have a picture from where you can see that the inversion layer literally keeps pollution trapped near the ground, significantly worsening the quality of the air. Now, because these are purely atmospheric phenomena, temperature inversions can indeed be thought of as producing exogenous variation in the quality of the air. They are also considered to be the best among the best meteorological predictors for air quality. And indeed, these phenomena are thought to be co-responsible for some of the most catastrophic pollution events in human history, such as the Great Smog of 1952 in London or the Donora Smog of 1948 in the US. So, of course, the existence of a causal relationship between air pollution and AMR has important implications for the cost-benefit analysis of air pollution control policies, because this means that the existing analyses in this sense do not take into account the impact that such policies have on antimicrobial resistance, and therefore the existing estimates of the economic cost of air pollution are likely to be just a lower bound. So for this reason, in this paper, after establishing causality, in a second step, I also perform a counterfactual analysis, estimating the impact of alternative air pollution control policies on AMR diffusion in the EU. And I also compare the effectiveness of these policies with more traditional interventions aimed at reducing antibiotic consumption.
And maybe it's not always obvious for an economist, and here we are already in a setting where you have economists and life scientists, but the exact chain of events. I can understand that if you have pollution all of a sudden, then you're attacked by microbes. Some of them are dangerous, and you might die. Now here, you're talking about something else, that I will not die, but that next time around, I may be more fragile with respect to, and that's because I will have taken some antibiotics, I guess, because then when people talk about antibiotic resistance, it's the fact that indeed you get used to this, and then so I think this, as I said, the diverse chain of events, you haven't yet talked about that, and I don't know. You will, yeah, but you might want to do it a bit earlier. I don't know.
Okay, yes. Yeah, indeed, I will talk about this later. Yeah, maybe I can mention this earlier in the presentation, but maybe let me proceed and then we go back to this point, because at some point in the paper, I try to distinguish these two channels. And So let me tell you how the paper locates into the existing literature. So we know that there exists a vast literature, health economics literature on the environmental determinants of health. Many papers in this literature have shown how air pollution indeed leads to increased mortality, hospitalization, and also increases in the incidence of certain diseases, especially respiratory and cardiovascular diseases. Some of these papers also specifically focus on children's health, because of course this is possibly the most vulnerable age group. But none of these papers has investigated the causal relationship indeed between air pollution and AMR instead. There is also an emerging literature studying antibiotics markets and the spread of AMR. Some papers indeed have showed how increases in antibiotic use causes increases in AMR rates. Some other of these papers instead evaluated the effectiveness of financial incentives given to the pharmaceutical companies to encourage research and development for novel antibiotics. But again, none of these papers has investigated instead the environmental determinants of AMR diffusion. And finally, there is a relatively recent and relatively thin non-economics literature on AMR and pollution. What papers in this literature typically do is they collect samples of air pollution in specific locations at specific points in time. They analyze them in the lab, and they found, as I mentioned, that indeed air pollution carries important quantities of antibiotic-resistant bacteria and genes. But of course, you can see that these papers are not able to quantify the impact of pollution on AMR at a more aggregate level. To the best of my knowledge, the only attempt in this sense has been done in the paper by Zhu et al. 2023. In this paper, authors collected information on PM2.5 concentration for several countries in the world and also on AMR. They studied the association between the two, and they found a positive correlation. But so, to the best of my knowledge, no causal estimates have been provided so far.
Maybe coming back to my question.
Yeah.
There is indeed this literature that says more antibiotic consumption means more AMR.
Mm-hmm.
Will your story be about pollution means more antibiotic consumption and therefore AMR, like these other people are saying, or is there another channel on top of that?
Both. So, and I'm trying here in this paper to also distinguish between these two channels. So, because indeed pollution increase, you may get sicker and consume more antibiotics, and therefore this affects AMR. But I'm also trying to distinguish this channel to what I call the direct channel that goes more through processes such as indeed evaporization and aerosolization and horizontal gene transfer. This is what I call the direct channel.
I think you may want to stress that because it's helpful and it puts the literature more in perspective.
Yeah, that's right. Yeah. Good point. Thank you. So before going a little bit more into the details of the analysis, let me tell you about the data collection process, and also let me give you some descriptive statistics. So for this paper, I needed to collect several types of data from several different sources. First of all, of course, I needed data on antibiotic resistance. I obtained this data from resistance map. Here, data are provided at the country and year level, and resistance is always expressed in percentage terms. This is because resistance is calculated as follows. The laboratories and hospitals part of the network, of the surveillance network, collect samples of bacteria, and then they test in the lab whether they're resistant or susceptible to different antibiotic classes. And eventually, resistance is calculated as the number of resistant isolates, resistant samples, over the total number of samples. Then, of course, I needed to also collect data on pollution, and particularly on fine particulate matter. For this, I used the data set made available by Ivan Donkelar et al. These researchers combined monitoring station data with satellite information, and they also use some sophisticated transportation model. But so what we really need to know here is that this is considered really the state-of-the-art data set for PM2.5. I also collect data for other pollutants, the four you can see on the slides. This is information provided by ground level monitoring station. And also this information is made available by the European Environmental Agency's Airbase database. I choose these pollutants because these are considered to be criteria pollutants in Europe, because indeed they are the most dangerous for human health. Then I also, of course, needed data on weather conditions, most importantly to build my temperature inversion instrument. For this, I use data on temperatures at altitude made available by the NASA's MERA database. So for each altitude, I have information on temperature, and each data point here, so these are gridded data in the sense that to each data point is associated a latitude and a longitude. And each data point is about 50 kilometers away from the next one. So here, to build my instruments, I needed to do two things. First of all, I needed to define temperature inversions. And second, I needed to find a way to aggregate this information at the country and year level. So let me explain this better with the help of another slide. So as for concern the definition of temperature inversion, I distinguish between surface inversions and inversions at any altitude. I say that a surface inversion occurs when temperature at the second lowest level in the atmosphere is higher than at the surface. I say instead that an inversion at any altitude occurs when the temperature at any level above the surface is higher than at the surface. So this is for what concerns the definition. As for the aggregation process, I proceed in a few steps. Here to simplify, let me say I proceed in two steps. First, and based on the existing literature, I count the number of inversions occurring in a year in each NUTS 3 region in Europe. So NUTS is a nomenclature used to divide the European territory in small geographical units, and NUTS 3 is the smallest unit in this nomenclature. Then, yes.
So as far as I understand, these inversions can last a day or a month, right? So why do you count the number of occurrences instead of the number of days? Because I guess if it's one day, I will not feel, I will sneeze one day and fine. If it's a week or two, I can start having deep irritation, feel sick, Why do you have number, not basically a count instead of intensity?
But actually what I'm doing is counting the days because the data, because the data are aggregated at the daily level.
Do you have the sequences, number of days in a row?
And I count, so when I count the number in a year, it means, it means that I'm counting the days in which these phenomena are. And this was based, by the way, on the existing literature.
But you have those that last more than X days, for instance?
Some can last just one day, some can last several days. I never heard about an inversion lasting 1 month, for example.
Well, I guess the smog of London was something very long, right?
Yes.
So
I have to check in my data what is the...
Can you distinguish between short and long? Could you add this as an intensity of treatment instead of just number of days of treatment?
Yes, I could do this. I'm not doing it now, but I can do this, yes.
So until then,
you may want to say, I count the number of inversion days
Yes.
during the year.
Yes. Yes. And then, yeah, okay, so this is for the first step. Then in a second step, I aggregate at the national level by considering weighted averages of these inversion frequencies, where their weights are given by the ratio between the NUTS3 population and the total country population. So in this way, I can still exploit the local population exposure to different frequencies and length of inversions. To give you an idea of the distribution of this phenomena across NUTS3 regions, so this refers to the first step, I have here a map. So this is, again, counting the number of surface inversions for the year 2019, which is the most recent in my
So we should move away from Brussels.
We need to go to Italy.
In Italy, they eat antibiotics like crazy, no?
I must say that though this is considering the total number of inversions, if I have also maps distinguishing between summer and winter, and you would see that the distribution is different.
Okay.
Yes.
So let me recap. We have PM 2.5 as one of the X variables. We have this as the
instrument,
the instruments, and we have antibiotic consumption as an outcome.
Antibiotic resistance as outcome.
So how do you measure antibiotic resistance?
So it's this is given, I'm gonna say, yeah, this is given, typically it's measured in percentage terms because what they do, so there is in Europe a surveillance network. Several laboratories and hospitals in different countries are part of this network. They collect samples of bacteria and they test whether they are resistant or susceptible to different classes of antibiotics.
That's country level, you're saying?
Yes.
So don't you
because
have
then
more local data?
No.
Hospitalizations per hospital or?
So I'm trying to find more granular data also because.
Or prescription of antibiotics at the NUTS3 level? Like you did for the opioid prescriptions?
It's maybe easier to find AMR data rather than
which data?
Resistance data rather than consumption data.
But the resistance is national, not NUTS3.
In this database, yes, I'm currently trying to find more granular data for AMR. For example, I know that there are some data, more local data for France on resistance, but this is still at the regional level. So
that's one.
That's one indeed, and that's only one country.
I'm saying it is not one. You say at the regional level.
Yes.
The region is a NUTS1 subdivision.
Yes, indeed.
Okay.
Yes. I had found also for the UK some more granular data, but it's not this easy.
And I guess you have density and population in each of these NUTS3 regions, right?
Yes. Indeed. Yeah, okay. So I will need in the analysis to also control for ground level weather conditions. For this, I use the information made available by the European GRC. These are also gridded data, and the aggregation is done in the same fashion as for the temperature inversion instruments. Then I will also use data on antibiotic consumption from resistance map. Resistance map is sourcing this data from the IQVA. So this is IQVA data. Here, consumption is expressed in terms of number of DDDs per 1,000 inhabitants. DDD means defined daily dose, and it's essentially the daily intake of a drugs for his main indication in adult. So it's basically consumption measured in terms of number of daily doses. Yes.
And that is at which level?
Still country level.
Still national.
But I3h, as far as I know, doesn't have more granular than this.
No, it's just because you have this super granular data to
It's
find
a
a way
pity.
to...
No, I agree with you. It's a pity not to be able to exploit such granular.
Exactly.
I agree with you, and that's why I'm trying to make a real effort to find more granular data for each country, but it's not super easy.
Antimicrobial resistance, I guess, is a very long-term process, whereas if you could find, you know,
the immediate
stuff with just the link between pollution and antibiotic consumption would be interesting if you could have that at an S3 level. This is what I'm thinking, you know.
Considering only consumption and resistance, though there is already work on this,
but the causal effect of pollution on antibiotic consumption would be quite interesting as such. I'm just saying, you started with, we have different channels, right, which have been explored, conjectural. I will do the first causal analysis. If you could have also messages on which channels seem to be more likely in terms of intensity, etc. Not a lab experiment, but behavioral changes caused by pollution. I think it would be great.
But you really have things to say about that.
See, I'm doing something along these lines.
But this being said, I think, although it's, of course, more data is always better, that, I think antibiotic consumption is very much the culture of a country. I think there are the things, since, you know, doctors are national, kind of thing. So I'm less worried about that one than others, because you want to control for the underlying.
We have this link, pollution kills.
I agree with you that this would be nice.
And here's if you want to fight antimicrobial resistance disease, this kind of particulates can be helpful. So don't think just in terms of medical things.
But she will, but okay. But I think it's important. I mean, the fact that these kind of questions are being asked, I think you want to put answers ready in your presentation.
So, and data for other controls are collected from the World Bank database. Okay, so let me show you a few descriptive statistics. Here I'm reporting for the main variable of interest, but I also have the full table if you want to have a look. So on average, the average resistance rate across European countries and for the period under analysis was 24.4%. Pollution was around 13.4 micrograms per cubic meter. This is the unit of measurement for PM2.5 concentration. And we consumed on average 8,600 DDDs every 1,000 inhabitants. But I can show you that there is a substantial variation for these.
Well, this suggests that indeed what really varies a lot is pollution.
Let me show you. So I have the descriptive stats by country.
Or the coefficient of variation, which is mean or standard deviation. That's what I was doing. If you go back to the previous, you can see that the standard deviation of pollution is bigger than for antibiotics, while the mean is tiny for pollution. So that was just my general point. You have a lot of variation of pollution, and then, of course, it may have to be the fact that you only have data on your country, that you have less, a lower coefficient of variation. But here it's very clear. The resistance is reasonably stable also relative to the pollution and consumption of antibiotics also. But you have a lot of variation of pollution. So.
Let me show you the descriptive statistics by country. Indeed, if we look at these three main variables of interest by country, we see that, for example, a country like Romania has about 5 times, so in a country like Romania, the resistance rate is about 5 times larger than in the country with the smallest resistance rate, which is the Denmark. If we look at pollution, we would see that a country like Bulgaria which is the most polluting country in the data set, is polluting about 4 times more than the least polluting country, which is Sweden. And finally, if we look at consumption, we see that a country like Greece is consuming about 3 times more antibiotics than the Netherlands, the least consuming country. Okay, so before proceeding, let me describe briefly how the paper is organized. So you can think about it as divided into 3 main parts, 3 main sections. In the first part, I provided the baseline analysis, and I also provide an heterogeneity analysis across different pathogen antibiotic combinations. I'm also planning to add to the analysis an heterogeneity analysis across age groups. This part is in progress, and we will see why in a moment. The second part instead contains mainly the counterfactual analysis, but indeed, before going to the counterfactual analysis, I'm including consumption in the analysis as well. And indeed, I will try to distinguish these direct and indirect channels. Finally, in the third part, I want to add a cost-benefit analysis for each of the policies under study in part 2. So, for part 1, so the main objective here is indeed to provide causal estimates for the association between pollution and AMR. To do this, I run the following Poisson model, where the dependent variable RCT is the average resistance in country C and ERT. PM2.5 is, of course, the average fine particulate matter concentration. And then I have a series of controls. For example, D contains demographic and health-related variables. OP contains the other pollutants. And finally, WCT contains ground level weather conditions. This is important to control for weather conditions on the ground in this type of studies because these weather conditions may be related, may be correlated with my instrument and have their own impact on resistance. Because of the strong time dependence of resistance, I'm also controlling on the right-hand side for resistance in country C in period T-1, so lagged resistance, essentially. Finally, in each regression, I control for country and year fixed effects. For what concerns the instruments, we already partly talked about this, but so I'm basically testing the robustness of my results to different instruments groups. What you need to know here is that I'm exploiting all the information at my disposal. So I'm considering the number of surface inversions, the number of inversions at any altitude. I'm distinguishing between winter and summer inversions because these phenomena exhibit clear seasonal patterns, and I'm also considering their intensity as given by the difference in temperature between the inversion layer and the layers below that one. For each instrument group, I interact by country dummies. This just means this is just a more flexible approach that allows inversions to have a different impact on pollution across countries. Okay, so these are the results from the baseline analysis. Each column in the table refers to one different instrument group. the main takeaway, I'm reporting here only the estimates for pollution and resistance. Again, I have the full table if you wish to have a look. So the main takeaway is that all my estimates are positive and mostly significant, and their magnitude should be interpreted as an elasticity. So if we look at column 1, for example, this means that a 1% increase in pollution leads to a 0.7% in antibiotic resistance. So this effect is quite sizable, and one reason why it looks say big is that before that, let me also talk about the heterogeneity analysis. So using the same specification, I also run the analysis by considering each combination of pathogen and antibiotics, and I find that indeed there is some heterogeneity across antibiotic pathogen pair. Now, here I find that, for example, the aminopregoside antibiotics are in general quite combined with any pathogen, are quite responsive to changes in pollution, especially if we look, for example, at panel B for the I3h. And also, if we look at the combination between these antibiotics and the pneumonia aeruginosa. I also find positive and significant estimates for the combination between this pathogen and ceftazimide. What I found also important here is panel H. So this is basically the methicillin resistant Staphylococcus aureus. And as you can see here also, estimates are quite sizable and significant. And I found this important because this is considered to be one of the top killer bacteria. So basically, this is essentially telling us that policies aimed at reducing pollution may help a great deal to reduce the spread of this killer bacterium. Yes?
So which of these affecting the lungs first or are airborne? Because the Staphylococcus, as I know, from what I understand, is more about contact. That's why it
I knew this. Sorry.
And I saw many of I3h something, but
I knew that the Enterococcus fecalis and faecium instead they are mostly by contact, while the others are more.
Pneumonia sounds transparent, but that's not significant, or not much. Even with a negative coefficient group.
Yeah, but it is here, for example.
Which one?
In panel E.
Yes, and that's what?
Pneumonia aeruginosa.
And the previous one, at the D was, what's called pneumonia? With
fluoroquinolone.
You're very good at this.
I practice a bit.
I would say for a humble economist, this goes faster. Also, there are these three control. I mean, so I think it would be nice to either focus first on a key thing and then go to robustness, or just this is a bit overwhelming.
Too much.
Yeah. I don't know the others, but me.
So in fact, it's the first time I include the heterogeneity in the presentation. That's why before I was tempted to jump because I was not used to have this part, but I thought it may be useful.
Already the previous slide, you had only 4 columns, one slide, and one of them it didn't work, and the other 3 it worked the same way. I was already thinking, okay, these are these 4 different approaches. Should we worry about one of them not being significant, or
No, because it's so often the case that different set of instruments they can predict better or worse.
Okay, because then of course why 4? Why showing 4? I mean there are a number of. I can see some readers saying, "Whoops, can you come back and explain a bit more detail?" That was my point.
Ask a small question.
Yeah, of course.
you control research? When you mentioned the health-related controls, do you control for hospital-acquired infections? The rate?
No, that's a good point, actually. And this is, yeah, so no, I'm controlling for health expenditure, but I don't have a measure, I don't have yet a measure of healthcare acquired infection.
But I think it exists. I think it's publicly available.
Yeah. I have to think about how to introduce this in the analysis, but this is something that I already had in mind. Yes, that's a good point. Yeah. And
so is a good, so again, what you could do here is with what is airborne, what is contact, and what I understand is contact is just as significant as airborne.
yeah, exactly.
Right?
So
Which would
the reason why I wanted to present this heterogeneity analysis today is to see whether there were more insights, especially on this part. Because, for example, what I read is that the E-facium is also mainly acquired by contact, and it may be surprising.
So does that reinforce the message that it's because people lock themselves inside that we exchange bacteria? It's not because of the pollutant who comes to my lungs with a small bacterium attached.
So what I had, so what I had in mind actually is a different mechanisms. So in order to try to find some explanation about this, I went back to the scientific literature, and I found some papers because this is happening for aminoglycoside antibiotics. I tried to find more information about that, and I found that these antibiotics are particularly not biodegradable, and therefore they would remain in the environment for longer. Because they would remain in the environment for longer, they're also more likely to remain into the air through again evaporation.
Or in the wastewaters.
Yeah, at least this is the explanation
Question.
And I'm doing a mess with the slide. Yes.
Yes, I thank you for presenting so far, and I like the heterogeneity analysis you did, because I think indeed it's good to see that airborne and contact are more or less the same. I just wanted to think that a further analysis that might be important is to what extent hospitals are implementing AMR measures, because there's quite a difference even in Belgium with the how do you say that, the countermeasures or to circumvent or to avoid AMR. I think that this might be also a very interesting one. For example, for your sample H on MRSA, I think, well, we know that this the most important source of environment where it's spread is hospitals, that the measurements or the countermeasures to spread this might be an interesting factor.
Yeah, I agree.
To include in your sample.
Do you think that by controlling in my analysis for healthcare expenditure at the country level, I can already pick at least part of it?
Yes, but I think you will have to go deeper.
Okay.
Not sure.
Okay.
Would anticipate. Yes.
Okay, thank
because
you.
Really, because you already see in Belgium as well, and there was a huge documentary in the panel, which is a Flemish Flemish, how do you say that, documentary? Yes, where they really looked at the VUB hospital, where they didn't implement, although all the audits showed that it was so important to control for AMR, that they didn't implement it so far. So I think on the national level, you don't have sufficient data. It might even be important to do on the regional level.
Yeah. Yeah, thanks. Yeah, thanks, Hilda.
Welcome.
Okay, so indeed, as I was mentioning before, the estimates we obtained from the baseline analysis look quite sizable. And one reason for this is that these estimates are likely to, in fact, capture the total impact of pollution on antibiotic resistance. This means, so why do I say total? Because the increase in pollution, the unexpected increase in pollution induced by temperature inversions are indeed also likely to make some people sicker and induce them to consume more antibiotics. If this is the case, then the coefficient associated with pollution in my baseline analysis would capture both the direct impact of pollution on AMR and also this indirect channel through increases in antibiotic consumption. Yes.
I understand that indeed.
So this is the second one is the mechanism you were pointing before, right?
That's right. And that one I understand. So I'm hit with these bacteria. I take antibiotics, and therefore, in the future, I am less resistant, and therefore the bacteria is more resistant. The direct impact of pollution, how does that work?
So this refers to the channels. So this would be the net, let's say, impact of the channel going through evaporization, aerosolization, and horizontal gene transfer. The reason why I was saying before I have to think, Siri, to go back to your point, I have to think how to fit the healthcare acquired infection in the analysis is because what I had in mind is the following. Suppose that because there is more pollution, you need to go to the hospital, and then in the hospital you catch, it is in the hospital that you catch the antibiotic resistant bacterium, then this would be still captured here. If instead you go to the hospital and you're given antibiotics, this would be captured here. So in order to isolate the channel you're pointing at, yeah, I have to think a little bit more.
Shishan is also raising his hand.
Yes. Hello.
Shishan, we don't hear you.
I guess you need to activate.
Yeah.
Yes.
Can you hear me?
Yes.
Yeah.
Yeah. Hi, Ilaria. Sorry, I was not here because I removed my wisdom teeth last week. So the question here is that in your direct channel, so if the pollution has the impact on the AMR on the human beings, so does it mean that the pollution also can affect the AMR of the animals? And would there a sub channel that so the pollution can affect the AMR of the animals, and there are some people consume more chickens, for example. There will be transfer of this AMR from animals to the human beings there.
Yeah. So the use of antibiotics in agricultural farming, in animal farming and agricultural activities is indeed an important driver of AMR2. I'm not currently looking at that in this paper. I may consider also adding this channel. And focusing on this is actually also something I have in mind for future research. But for the moment, I'm not considering animals in this paper.
Yeah, because in your heterogeneous effect there, there are 4 different types of the antibiotics there, and I'm wondering that if any one of them has been widely used in the animal field, maybe this is also caused
So the data I'm considering, this is the antibiotic consumption by humans and AMR in humans.
Okay, yeah.
I think the point is if the same type of antibiotics is used both by cattle and humans, maybe the effect is bigger. That's what I understand as being the question. Is that correct?
Yeah.
Sophia has a question.
Yeah. Okay. Yeah, Sophia.
Hi. Hello. Hi, Ilaria. Can you hear me well?
Yes, I can.
Oh, perfect. Thank you so much for your presentation. It's really interesting. I was just wondering if one of the things that was discussed is precisely because you have data at the national level, and I tend to agree that health expenditures might not be the type of measure that might be more appropriate for what you're trying to measure. So I was wondering if you could find any information on, for instance, types of tests that can be performed to understand if there's really a bacterial infection or not.
Sorry, I didn't understand which type of data?
On tests that can be performed to detect if it's a bacterial infection.
Oh, but this is the type of the type of information they use to calculate my outcome variable.
So you're using the tests,
yes,
that are performed.
Yes, isolate tested. Yes.
So then your measure that you have is based on a national level, like all the tests that were performed from which there was indication.
Yeah, exactly. Yes. In each country and in each year. Yeah, exactly. Yes.
I mean, it's very likely that in many cases you are not tested. So you're just given an antibiotics without being tested for having a bacterial infection.
Yes.
And we're trying to find some variations. So for instance, in Denmark, it's much more likely to have this type of test before giving you an antibiotic. My understanding, for instance, in Belgium is that it's extremely heterogeneous, actually. So then forget about that part. I was also thinking about the acquired infections in hospital, because that's the type of information that you normally have at the hospital level. But many of this, it's also captured by going to the GP and not ending up in the hospital. So, okay, I'll try to think then about another one. Sure. Okay, thank you so much.
Thank you. Okay, so direct and indirect channels. And okay, so I was saying, because I have data on antibiotic consumption, I can introduce this variable into the analysis and take antibiotic use explicitly into account. I'm also instrumenting for antibiotic use. Okay.
You have half an hour.
Very bad. Okay. So okay, let me speed up a little bit. So these are the results from the analysis that considers together both pollution and antibiotic consumption. The main takeaway from this table is that indeed, once we add antibiotic use, the coefficient associated with pollution is more than half, consistent with the idea that the estimates from the baseline analysis capture both these channels. However, the coefficient associated with pollution remains positive and statistically significant. And also, if we compare the magnitude of the coefficient pollution and antibiotic use, we see that pollution is not only an important driver of AMR, but actually it's almost as important as antibiotic consumption. So the coefficient for pollution is smaller, but it's still quite important in magnitude if compared to antibiotic use. So,
sorry to be off topic now, but why Poisson model?
Because I didn't say this in the paper. This is not the only one model I have. I have the Poisson, I have a fractional logic model, and I have also a linear model. So in a sense, I'm also testing robustness across different specifications. The Poisson is the one I prefer because my outcome variable can only be positive.
The Poisson is continuous or is it a count?
It's a count typically.
This is why.
But I have, yeah.
Since it's a percentage, your dependent variable.
Yeah, that's the idea.
The logic with the other one you said.
The fractional logic is also appropriate for this.
Isn't it more intuitive than the Poisson?
You think it's more intuitive?
I'm asking. Did you try the different methods of presenting and?
Okay, so these results are those that I will use for the counterfactual analysis, and let me finally get there. Actually, no, before that, let me just briefly talk a little bit about the EU air pollution regulation and the objectives that the EU has for the future. So we can say that the EU has made effort to address air quality issues since more than 40 years now, the most recent piece of regulation being the 2008 Directive on Ambient Air Quality and Cleaner Air Quality. Now, more recently, the EU has decided to basically further strengthen the existing standards to make them more closely aligned with the latest WHO recommendation. So what we really care about for this paper is that the new rules that the EU wants to impose would set the annual limit value for PM2.5 at 10 micrograms per cubic meter, while the latest WHO recommendation set this limit at 5. Another objective that the EU currently has is to reduce antibiotic consumption by 20% in each country by the year 2030. So these are the three objectives that I focus on in my analysis. And here's the results. I'm reporting for a subset of countries. Again, I have the full table in case you're interested to see results for more countries. So in this table, the first column here reports for each country the resistance rate for the observed level of pollution, consumption, and all the remaining control variables. So basically, it's resistance in the status quo. The following two, instead, consider the impact of the two pollution policies. So each of these numbers basically is telling us what would have been the resistance rate in each country had the EU kept PM2.5 at 10 during the period under analysis. Or what is the resistance level had the EU kept PM2.5 at 5 during the period under analysis. The following column considers instead antibiotic consumption only. So what would have been resistance had the EU had antibiotic use been 20% lower in each country during the period under analysis. And finally, the last two columns are considering the joint impact of each of the two pollution policy with the antibiotic consumption policy. So, of course, here results will depend on the situation, on the specific situation of each country. For example, if we consider a country like France, which is a relatively low polluter in Europe and a relatively keen antibiotic consumer, we see that indeed the impact of the consumption policy would be larger than the impact of the pollution policy. If we consider instead a country like Bulgaria, which is a high polluter in Europe, we see instead that the impact of the pollution policy is larger than the impact of the consumption policy.
Only a bit larger, no?
Sorry?
Only a bit larger, or not?
For Bulgaria, it's not this small, I find, no? Because it's 26.57 versus 30.56.
Okay. So that, well,
it's a bit smaller for
+6, and consumption policy it's +5, no? Or not?
Yeah.
6.5. Oh. But in any case, France, as you say, it's a smaller one, but okay.
And obviously the joint impact of the two, so the two policies together would have the largest impact.
So you're not calculating thresholds, it's always elasticities in the regressions.
Yes.
So when you go from 10 to 5, it's just because you multiply the effort by 2. You have 2 times elasticity instead of 1 time. Ah, no, it's from the level.
So consider you estimate the
5.
coefficient, that's an elasticity, and then you just replace for the threshold that you want in pollution.
So you estimate nonlinear effects, thresholds?
I could do that.
That I think would be cool.
Yes.
Because if you could identify places where you see a big pickup.
Yes, indeed. It's something that I had in mind to also add as an extension to the paper, indeed. Because I guess also this is important for external validity, right?
And compared to the typical numbers we see in newspapers, they say, oh, the World Health Organization says it should be this number. But, you know, what would be the impact of going from a moderate reduction to a big reduction? Is it where the things happen or not?
Yeah.
So if you just have one elasticity, we don't really know if it's the average or if you can estimate a threshold.
Yeah. Yeah, this is something I wanted to do. Yes.
By the way, I guess there is this, in a sense, well, first of all, in terms of looking at countries, I think an interesting one is Finland, which is already doing everything right.
Indeed.
So you cannot, there's nothing to do there.
Indeed, it's not binding.
On the other hand, when you look at pollution versus consumption, you know, moving away from pollution may mean that you completely have to change your industrial structure or your housing structure.
Exactly.
While consumption could be easier. Because the good news is that you say that, okay, already thanks to consumption, I can go that far, but you still need, in some countries, like Bulgaria, you could go quite far. Okay, maybe.
Well,
and Greece seems strange for that, I find.
For Greece?
Greece seems strange because I don't know. I don't think this is a very industrial country on average. So
I'll double check that. Yeah, maybe it would be more intuitive.
Because then I would have expected that they consume too much and therefore the impact of consumption.
I'm sure the numbers are right, but I will check why this is happening.
And could you replace delta C equals minus 20% to going from the initial level to Denmark or to Finland?
Yes, I can do that.
It would speak more, I think, because then you have both with PMW.
You say the same for everyone, like why? So
exactly.
I have an exercise for this, but first I wanted to say something else. It's true that the stricter the pollution standards, the more difficult can be for countries to implement. What I can say here is, okay, I'm considering a policy that the EU wants to impose. And how they're going to do this is beyond the scope of this paper. But they want to do it, so...
Yeah, but that's exactly the point. Let's have a target of PM 2.5 of boom. Okay, so there you count, oh, I'm super polluted, I go to 10,
yes,
big effort.
Definitely.
But with delta C equals minus 20%,
It's a different type of thing.
Exactly.
Indeed, for this reason, I'm also proposing this exercise. That is, I'm now asking for each country what would be the reduction in. This was one of the first comments I got when I started to work on this paper. For this reason, I'm proposing this exercise where I'm asking, okay, for each country, what would be the reduction in antibiotic consumption needed to obtain the same reduction in resistance as under each two of the pollution policies. So again, in this table, the first three columns are exactly the same as before, but now indeed in the last two, we have the reduction in consumption needed to achieve the same objectives as the pollution policies. So at the end of the day, I find that in Europe, the new rules that the EU wants to impose would decrease antibiotic resistance by 2.33 percentage points. And in order to obtain the same reduction in resistance, we would need to reduce antibiotic consumption by about 23%. Which is approximately the objective that the EU has for the year 2030. So it seems reasonable. Okay, now part three, what I would like to do is provide a cost-benefit analysis for each of the policies that I consider so far. So to do this, as a first step, I need to be able to quantify the health burden associated with resistant infection. What I would like to have is a measure of mortality and also dailies. For sure, you all know that daily means disability-adjusted life year, and this is obtained by taking the sum between the years lived with disability and the years of life lost due to premature mortality. So to do this, so I found that there are not many papers in the literature doing this. I have chosen to obtain these measures of health burden by following the method developed by Cassini and colleague in this paper dated 2016. So basically, they develop a model that they call outcome tree that allows you to derive a number of deaths and dailies associated with resistance infections by imputing the number of resistant infections. So in order to be able to use this model, I need to have data on the incidence of AMR infections by age group, gender, and pathogen antibiotic pairs. I'm currently really these days talking to the ECDC, the European Center for Disease Control and Prevention, to obtain this data. So let me explain you briefly how this model works. So this is what they call the outcome tree. So essentially, once you have the number of resistant infections, you can input into the model, and this allows you. So basically, this number is telling you that among all the resistance infections, a percentage between 9.4 and 20.3% leads to death. They also distinguish between short-run and long-term consequences. So, for example, the yellow box represents the short-term consequences. They distinguish between complicated and uncomplicated infections. And so, if we want, for example, to measure the number of years lived with disability, we take the total number of infections. We know that a percentage that's between 34 and 47 is complicated, for example, and then we multiply this number by the DW, that is the disability weight, and the duration, that is the amount of time you spend in hospital, here expressed in years. The same is true for long-term consequences. So we know that, for example, 58.4% of the patients will have a physical impairment in the long run, And if we want to calculate the years lived with disability, we have to multiply the number, so the number of infections by 58.4%, and then by the disability weight, and for duration we have the remaining life expectancy. So you see why it's important to have data by age group, because obviously the remaining life expectancy depends on the age of the patient. And the same is true if we want to calculate the years of life lost due to disability. These numbers, all the percentages, the disability weights, all the figures you see in this model are extracted, are found by the authors through a very, very extensive literature review, and also based on expert consultation. This is what they call the baseline model. Building on this, they also provide some figures for specific pathogen-antibiotic combination.
The characters a bit big.
Sorry?
No, we're just kidding.
Yeah, it's not readable.
It's very small. Yeah, sorry, I didn't expect this. But yeah, so basically here each column refers to one different pathogen antibiotic combination, and again it's based on extensive literature review. And so once once I have a measure of mortality and dailies, I can again attach a monetary value to it, and this allows me to also calculate the economic burden associated with resistant infections. I'm also going to consider other type of costs, healthcare costs, which are typically measured by taking the product between the time spent in hospital and the average cost per hospital day in each country, and also labor market costs, which are often calculated by taking the duration of the work forgone due to illness by the national average wage. So, to conclude, this paper shows that pollution is indeed an important contributor to the spread of AMR in Europe. Its role is almost as important as the one of antibiotic consumption in driving rising AMR rates. And so all these analyses tells us that stricter pollution standards would help contain the spread of resistance and limit also, for example, the diffusion of some top killer bacteria such as medicinally resistant Staphylococcus aureus.
Thanks a lot.
Thank you.